and philosophy. Got it. This will be more about history and philosophy, I think, rather than hard data. But there are some data points later on. So I'll start with the scene where all this story takes place, down along the Red River in Lake Texoma. The Red River is dammed right by the town of Willis, downstream from that one a little bit. And here is Denison Dam. And that Red River forms the boundary between Oklahoma and Texas, eventually empties into the Mississippi and into the Gulf. So that's what Denison Dam looks like. And Denison Dam was really one of the major collecting sites for Teague Self and all of his the students that worked on Nematobothrium and buffalo fish. Here's a little bit of a blow up to the of the whole study area, and you can see that Lake Texoma is highly branched. It's one of the most branching lakes really in North America. 140 square miles. We'll talk a little bit about, or maybe mention, the Tishomingo National Wildlife Refuge, which is mainly a big lake and marshes up in near Tishomingo. So this is the whole area, and eventually we'll mention not only the people but all the study sites that Dr. Self and his some of his associates worked with, and those include much really of southern Oklahoma. Here are the players. Teague Self in his younger years. That's him in the hat. That's George Moore from Oklahoma State University. This is a picture back from the 50s. This is the picture that's posted at the in the main lodge there at the University of Oklahoma Biological Station. Carl Riggs started the UOBS in 1949. He and a number of students spent the summer on a boat, a big houseboat, studying fish in Lake Texoma. And very soon thereafter, Norman Brillhart donated the land and the building that built UOBS. The fish players are buffalo fish, Ectobius, a couple of species, the bigmouth and the smallmouth buffalo. The only person to ever well, first of all, this work took place from the early 50s through at least the mid 60s. So about 15 years worth of research, field work, and experimental laboratory infections attempted. So over that time period, the only person to really receive a degree from working on Nematobothrium was Lou Peters, who finished his career, or actually had most of his career, at the University of Northern Michigan. And Clarence Davis was, I knew Lou Peters personally. I also knew Clarence Davis. He was evidently on leave from Southern University in Baton Rouge, a traditionally Black university. And I've tried to get a picture of Clarence, and I remember him well. A picture of Clarence and any information about his subsequent experience, and nobody down there would respond to either my paper letters or my emails. Charlie Blevins I never met. He was a commercial fisherman, and we'll talk about him a little bit later on. Back in the 1950s at UOBS, that main building, this is what the Parasitology Lab looked like. This is what it looked like when I took parasitology there. 8 weeks of class every day for 8 weeks at UOBS, and this was the lab building. For those of you that may have been to the field station down there recently, that lab has been converted into a recreation room. So it's a place for ping pong. There may be a pool table in there too. So the situation is these fishes have worms in their ovaries, and the worms are really strange trematodes. And the scientist wants to know how the worms get into buffalo fish ovaries. The worm eggs are shed when the fishes spawn and the worms disintegrate, releasing the eggs. So really a fairly short time period during the year, late spring and summer, is when the massive numbers of eggs are actually shed into this very large lake texoma. The worm is now known as Nematobothrium texomensis, and the important part really of this talk is that it's the didymozoid trematode, and virtually all of these are in marine fishes. The life cycles are not known, but Louvard et al., and I'm going to talk about that paper, which is a 2022 paper from IJP. I'll show you some pictures from that a little bit later on, but the life cycles are generally not known at all. In fact, this is a very mysterious group of parasitic worms. Here's an excerpt from my book that comes out in August. The first chapter of that really is all about Teague Self's obsession with this worm and his attempts, and what that obsession taught him about parasites and about some of the larger questions in life. First report is in scientific literature. It's from the printed program, and this is kind of important. Printed program distributed at 1955 meeting of ASP. ASP is typical, holding annual meetings, publishing a journal. And for those of you that might not know it, the word "the" is part of the official title of the Journal of Parasitology, in which scientists can present the research results. Meeting was held at the Dinkler Plaza. That building was razed in 1972, but back in 1955, buses took ASP members and those who were at the meetings over to the biology labs at Emory University, where on the afternoon of December 29th, Alan McIntosh and Teague Self presented paper number 83, Nematobothrium texomensis, new species from the freshwater fish. New species, part of that title is important because it's standard abbreviation for new species. This is for my non-parasitologist readers. Some of you may know that Alan McIntosh from USDA was really a world-renowned expert on trematodes, and Self, when he finally tried to figure out what that worm was, then he sent specimens to Alan McIntosh, and McIntosh agreed that it was a trematode. Now, here's the abstract from that program, and it's important to know that back in 1955, the programs were actually printed and incorporated as part of the journal. So this is not like it is today, but back then the program functioned then as an official journal publication. So this one is about estimated. This is the original species description, and there are no surviving figures from this because they were all shown at the time and probably on glass plate slides, if I remember the lab at the time. This thing is 8 and 9 feet long, about the size of a pencil lead. So if you can put that kind of in your mind, that's what that trematode is, wound up in the ovaries of buffalo fish. The second report of this thing in the literature was from a paper that Self and a graduate student, James Campbell, published in 1955 in the, actually I found 56 in the Transactions of the American Microscopical Society. This is actually would be the equivalent of class project from field parasitology at Cedar Point, because it was all of the students at UOBS that summer that collected fish and went through them. So here's what it looked like. 2 specimens over 17 feet combined length. So this is a worm that is very, very long, that is entwined in the ovaries. Now, a couple of what I would call museum finds that are that sort of led me to investigate what I knew about from graduate school, but really didn't know about. And these are field notebooks that Scott Gardner refers to as a codex, codices, because they sort of function that way. And there are really over 360 pages of this kind of field note writing, and I'll talk about those things in a little bit. So here's what the Miracidium looked like to Teague Self. This is one of his drawings, and here he's talking about Lou Peters also fed some of these eggs to the snails, a couple of different species of snails, and they pass through the gut without hatching. So one of the obvious things you do trying to solve a life cycle with trematodes is put them in the water with snails where those eggs hatch and penetrate, larvae penetrate, or feed them to the snails where they hatch and penetrate the gut. So, but this is where really the information I'm talking about today comes from. That's next page. There's another page. He actually got these pictures by forcing the eggs, forcing the Miracidia out of the eggs with pressure. And what he discovered when he did that was he was able to figure out their structure a little bit, and but they blew up and died very quickly. There is really only one definitive scientific publication on Nematobothrium texomensis, and this is it. And you can see that these diagrams come really from Lou Peters and Teague Self's pictures from their field notes. That's the Miracidium. Here are the masses of eggs, hatched eggs. Now, the reproductive structures which really define this are pretty complicated and very sort of atypical of trematodes. And I assumed from looking at those published figures that they were actually able to decipher the male system after tracing the female system, and especially the uterus, which is pretty easy to figure out from the specimens. And it's from the permanent slides, and it's really the only one of those internal structures that's very obvious. Those are the pictures that ended up being reproduced more or less in Gerald Smith's How to Know the Trematodes. And so there's the reference in there of those figures. Here's what the specimens actually look like, and thanks to Gabor for taking those photographs. This is a specimen from the Harold W. Manter collection 30492. I've put a circle around that particular part of the worms. So what you're seeing, all of this stuff here, you can see the uterus filled with eggs. That's the structure. The only thing that I saw in all of those worms that looked even halfway familiar, and because the testes are described differently than they look like here, I have no idea what those structures are. So it would be a pretty good idea for some undergraduate looking for a nice project, get out all of those Teague Self specimens, remount some of them, do a detailed anatomical study of them. Uterus, however, is pretty easy to figure out, and there it is. This part right here, this clear part, is part of the digestive crura. So, and there's a drawing really of the uterus of eggs. This is the stuff, 7 or 8 feet of these kinds of worm pieces get liberated into Lake Texoma during spawning. Here's a little bit of a slight digression. This is actually the original description of the genus Nematobothrium by Van Beneden in 1858. And you can actually pull up that publication and scan through. What you're seeing here is a screen clip from that. You can't really copy it, but you can do the screen clips. And here are some of his diagrams of the thing. I won't really go into his in great detail, but this is what I am interpreting as his description of the genus, and that's my translation of it, going back to high school French, still incompletely studied. And he went ahead and published that original generic description in that monograph, without really knowing which end of the body was anterior, which was posterior. So now Omar Amin, however, also then did a study of catastomid fishes in Arizona, and pulled up, he gave it, pulled up some specimens and gave us some data on occurrence. Here's a table out of that particular study showing the prevalence of that worm in different species and in different locations. So something was actually going on in Apache Lake, and that allowed that worm to complete its life cycle. Now, what about the life cycle? And I'm showing you really a copy of, or a picture of Matt Bullock's iron wheel, because that's what I think of when I get into efforts to solve that life cycle. Here's a typical diagram of typical trematode life cycle. This comes to mind immediately. Every parasitologist could draw this thing from memory. The important part, however, is that this particular diagram also represents the expectations and the ideas that drive your attempts to solve a life cycle. So the expectations, which are Matt Bullock's iron wheel, they're really the scheme of things that in your mind becomes the way things are. So that's why you begin, if you're a parasitologist, by dealing with eggs and mollusks. And in our minds, then, we put animals on those boxes. And so this is really what's going through the mind, the pictures of the people that worked on this farm. Here are some excerpts from the codices down here, where whoever was writing this set of field notes, and I'll come back to that question here in a minute, exposed some snails to these ova and checked the feces of those snails. And the question is, did the eggs hatch? And little hatching occurred, a few viable eggs and no evidence of hatching. They also fed them to fingernail clams, and in addition to these two snail genera, and no definitive evidence that the clams had eaten, fluke evidence. And then they picked up some of these and tried to feed them directly to the fish, and those things, of course, never took. And those were difficult things to do. The second codex looks something like this, and it's written largely in first person, but there are no names and no initials in here that would allow me to figure out who actually wrote this. And the one surviving person that I was able to find, who was in grad school when I was there, was a person named Barbara Neese, who is now retired from a faculty position at the University of Oklahoma Medical School. We had a little bit of an email conversation, and then no more answers from Barbara. But some of these dates, for example, April 1st, 1963, that's when I was working for Dr. Self on my doctoral work in the Cheyenne Bottoms. So this is another codex. And I'm going to get to the point of the number of people who actually ended up being mentioned in these codices. The name Charlie Blevins shows up in places, and I'll come back to Charlie here in a minute. But the handwriting samples, this is obviously Teague Self, and Karen was his secretary. He was secretary to the Oklahoma Academy of Sciences, and in addition to the Nematobothrium grant, they also had a very large grant to send scholars out to small town Oklahoma to give programs and sort of deliver intellectual and cultural experiences. And Karen ended up being the secretary that handled really much of the administration of that particular grant. So she saw a lot of Teague Self's writing, and I saw a lot of it on my dissertation. So that's obviously Teague Self's. This was Clarence Davis's writing, and he initialed a lot of his. This is Lou Peters' writing because of the dates and what they ended up doing. This whole codex, 100 pages of it, in first person. I have no idea who actually wrote that. Here are some data from those field notebooks. I went through those, but I did not extract all of the data because there are pages and pages of these kinds of entries. April 22nd, 1963, 56 set of ovaries from Charlie Blevins. Charlie Blevins was a commercial fisherman who fished in Lake Texoma and would set a mile of gill net. And so that's how he collected that much stuff. And evidently, they, Dr. Self set up a relationship with Charlie Blevins so that when Blevins went through those fish from his nets, he would just save all of the ovaries and pass them along to whoever was working for Self at the time. So 56, and there were several of these kinds of entries. So Charlie Blevins ended up giving those folks several hundred sets of ovaries from buffalo fish. And these are the kinds of entries for every one of those sets of ovaries that you'll find. So number 21, whatever that was, the ovaries weighed 200 grams, and extremely long worms in both ovaries. And a month later, here's another 27 sets from Charlie's Blevins. And again, this gives you a sense of the prevalence of that. Now, do the Miracidia hatch in water and then penetrate the host? And here's an example from the attempt to figure that out from the codices. Miracidia had emerged from the shells, however, movement after hatching ceased either immediately by at least 5 minutes, and the Miracidia showed great hyperosmotic stress. So that's the kind of thing that tells you immediately that those eggs must be eaten by something that that then results in penetration, first intermediate host of some kind. Now, I'm going to digress a little bit and talk about the extent of this work over that 15-year period, and doing that by going through those codices and listing the names that are actually mentioned. And they don't necessarily always tell the roles of those people to various extent, but some of them do. Howard Clemens, for example, opened up. Howard Clemens was taught invertebrate zoology at OU. I remember him well. He's now deceased. I remember him well at Self's graduation, who he said to me, I'm sorry, Self's retirement, who sort of pulled me aside and said, You know, when I looked at my first paycheck from OU, I said, this is not going to get it. And I took out a loan and bought some rental property. And every time I could, I bought more and more rental property. So Howard Clemens became a landlord in Norman, Oklahoma. He also acquired a fish hatchery near the town of Noble, and that's where he did um, endocrinological experiments on channel catfish and tilapia. And he is largely responsible for the techniques for cultivation of channel cats and tilapia with his work in the 50s and early 60s. Lou Peters, like I said, is the only one who ever got a graduate degree. Mr. Russell, and it's only mentioned as Mr. Russell, was one of the commercial fishermen. John Fox was the zoo department storeroom guy at OU, consumed lab alcohol. Dan Harlow was a master's student with Dr. Self. He's the reason that I'm a parasitologist. When I went back to graduate school in 1960, actually the summer of 1960, Dan ended up in some of my classes, and he was one of Sieg Self's students. And for some reason or another, he would come over to our little cottage where Karen and I lived, and Karen would make cookies, and Dan would always bring along a bottle of Chivas Regals. So we would sit there and drink scotch and eat chocolate chip cookies and talk science. And he was studying the Helminth communities of pectoral sandpipers. And he was asking the question, do pectoral sandpipers bring Helminth parasites into North America in the spring and summer? And then do they leave with different sets of Helminth parasites after nesting in Canada? So he was really studying the intercontinental movement of Helminth parasites. And at the time, I thought that was really a powerful, conceptually powerful question that involved some seemingly straightforward and simple work that gave Dan this broad and rich repertoire of transferable skills. And after really dealing with Dan for a couple of years, I said, I'm going to be a parasitologist. James Sublette was an international expert on chironomid flies, and he was a faculty member at Eastern New Mexico State. And he became involved because Self thought maybe that chironomids were intermediate hosts for buffalo fish. And during those experiments, Self did some aquarium studies on the feeding behavior of buffalo fish and discovered that they would pick up a mouthful of debris and spit it out into the water and then pluck out any invertebrates that were in there, which was kind of fun. John Makowitz, some of you may remember as an expert on the helminths, particularly the cestodes of catastoman fishes. More names. Dr. Valentine had no idea who that was, but he was obviously helped Self and those folks identify some of their bivalves. And where did they collect? Buncombe Creek Arm is the one that's right there next to UOBS, but all of these other places, if you actually did a Google Maps search, you'd find that they cover much of southern Oklahoma, in particular the areas up near the Wichita Mountains, and as far north as Chickasha and the Washita River that also drains into Lake Texoma. And study sites continue really. What kinds of mollusks did they use in their work? A whole suite of them. And again, remember that these are names that were put on all of those mollusks in the 50s and the 60s, well before any molecular work let us know about snail diversity. So these are all snails, and some of them were really collected in Wisconsin and Michigan and brought down to Oklahoma by Lou Peters, a whole diversity of clams from southern Oklahoma. Now the question, what has modern science told us about didymozoids and especially their life histories? And this is, again, as far as I can determine, the definitive paper on didymozoid life cycles. And this is in IJP, and y'all can get this paper as a free download eventually if you want to go looking for it. This is the Louvard et al. Paper, and one of the co-authors then is Thomas Krebs, who again, some of you may know. First elucidation of a didymozoid life cycle. And what they did was conduct a major survey of mollusks, including bivalves and fishes from the Australian region. And they did this in sort of Janine Cairo ways, and that is by getting fish from commercial fishermen, from sport fishermen, from every source that they possibly could get them. And they dissected everything and saved everything and did molecular screening with ITS2, with 28S, and with COX-1 genes. And they screened all of this stuff. And if you get that paper and go into the tables, you'll find pages and pages and pages of GenBank numbers and where they came from. So out of those nearly 200 specimens of Arca trapezia, and these are arc shells with taxodont teeth and very heavy shells and thick shells. They never observed natural. These are cystiferous cercaria that are hemioroids, and we have some of those. I think that Halopegus in local frogs has those cystiferous cercaria, but they never found any, and they kept them in aquaria, and nothing ever emerged naturally. So they did all of this stuff with dissection. However, here's what the Metacercaria looked like. This is the cyst, and that's the sort of tail, and that's the sporocyst that contains all of those immatures. So this is what the dissected stuff looks like from the clam. All of the, and this is important, I think, all of the fish specimens that they collected were embedded in the various tissues. And they, this is an example of the Elops hawaiiensis. These fish are really coastal predators, and they can get maybe a foot, 2 foot, 2 feet long. But again, in all of the fish they got, everything, all of these adult worms were encysted and embedded in the tissues, which tells you that perhaps even though these are predatory fish eating some kind of intermediate host, then something may eat those, or those fish have to die before those eggs are released into the environment. So this particular paper and that staggering amount of laboratory and field work represents a truly major accomplishment and step toward a resolution of the life cycle. But it's a long way from the kind of life cycle understanding that we generally use when we think of things like frog lung flukes or fasciole hepatica or dicrocelium, something like that. Nevertheless, that is a major step toward doing that with new technology. Again, 60 years after all of the self work. There have been a number of pelagic fauna, including things like ketognaths that have been proposed as second and third intermediate hosts, but we don't really know what those are yet. So here are some, now to get to the rather philosophical part, some evolutionary thought experiments. What factors have driven acquisition of this highly atypical body structure and host use and life cycles among one group of trematodes, and particularly the Nematobothriids? And if you get into the Louvard paper, you will discover that they've also generated some phylogenies based on didymozoid sequences, and the discovery or the realization that there's not a lot of monophyly involved in this whole group, and there's a staggering amount of work still to be done to sort out the didymozoidy. So if anybody who needs a lifetime worth of research on parasites and as just one Powerball, as we used to say at Barry's, the didymozoids would be the one for you. So what factors have driven acquisition of the highly atypical body structure? And if you're so inclined to kind of reconstruct typical Barry's bar talk from the 70s, you might conjure up the most extreme cases of atypical body structure and host use that we see from the daily news today. What are the life lessons from all of this Nematobothrium work? And that what we're really asking in this case is, what do we learn from doing all of this work? What do we learn from our research subjects in addition to what we learned about them? And my contention is that in the long run, what we learn about them is really subordinate to our careers and to our contributions as scientists from what we learn from our research subjects, what they teach us about. So the Nematobothrium texomensis life lessons. First of all, what happens in the natural world really has no necessary connection to your beliefs, your desires, or to what others have said. And I will tell you that every time I log off of my email, my Hotmail account, and see the in Microsoft News, I think about this over and over and over again, that there's so much going on in the natural world that has no connection to your beliefs, your desires. When DeSantis decides to eliminate the term climate change from Florida school curricula and discussions in the legislature, that's a perfectly good example of that. Your expectations can hinder a search for the truth, and that was clearly the case with Nematobothrium texomensis. And so the other thing we might ask is, how many other situations are we in which our expectations can hinder a search for the truth? And finally, really simple problems can have solutions that defy the finest mind. You can bring up all of today's hot button political words, and those are seemingly simple problems that are truly complex, and they are their solutions are defying the finest minds. The only pictures that I could find of Teague Self in all of my collection of scanned stuff at UOBS was this one. And he's standing next to one of those apartment buildings. And of course, you'll recognize this as Tim Runke, one of the parasitology graduate students at the time. And so those are the life lessons. If you float those life lessons out in front of a bunch of parasitologists, especially ones with any research experience, they're likely to say, duh, what's so special about those life lessons? We knew that all along. We learned them. And so here are some parasitologists for you that just as some of you may know. This one is Brent Nickel, faculty member at UNL for years and years and years, decades, former editor of the Journal of Parasitology. Among the ones I know, this is Tammy Percival at the time, now Tammy Percival Cook, recruited out of Biology 101 to come into field parasitology. She's now Tammy Percival Cook, master's at UNL, PhD at Texas A&M. She's now a faculty member at Sam Houston State University and former president of the American Society of Parasitologists. Bill Mosier, recruited out of freshman classes, now curator of inverts at the Smithsonian. Dennis Richardson, one of Brent Nichols' doctoral students, now faculty member at Quinnipiac. Dan Halliday, recruited into field parasite, now employed by Alaska Game and Fish. Skip Sterner, former museum collections curator. Scott Snyder, recruited out of introductory biology, master's degree at Wake Forest, PhD at UNL, now vice chancellor at University of North Dakota. Mike Barger, recruited out of freshman biology into field parasite, master's degree with Brent Nickel, PhD with Gerald Esch, now dean at Stevens College. Rich Clopton, recruited out of freshman zoology, PhD in UNL. Peru State professor, editor, now editor of Journal of Parasitology. Deb Clopton, excuse me just a second. I will. I'll call you back later. I'm in a meeting. Bye. Karen's not here to answer the phone, so. Laura Krebs, one of my former students, Nate Cody, I've lost track of. Those folks, if you talk to them about those life lessons in comparison to the general public, and now we see the real challenge for scientists who understand those life lessons and the others to carry those messages and to try to make the general public understand how important those life lessons are and how they apply to situations far beyond the research laboratory. So I need to acknowledge Scott and the Harold Manter Lab archives, Gabor for taking the pictures, all of those deceased and otherwise incommunicative folks who worked on that project, the UOBS, Charlie Blevins, who delivered all of those hundreds of ovaries to the folks that who were working on this project. And I'd be happy to try to answer any questions that you may have. So I'm going to go ahead and stop sharing. And it looks like there's a chat.
Thanks, John. That's cool. Let's see who said something. Sebastian said he had to leave. Dennis, let's see, DF. I don't know who DF is. Said, well, oh, Dennis Feely said, well done. So that's cool.
Thanks.
Yeah, really just riveting to me. For me, this is the kind of stuff that makes everything happen. You know, it makes us know why we're here, makes us know why we are, why we spend the time up here to take care of these books, to take care of the specimens, to take the records and put them online, to do all these things, to make this stuff available for the future. And
Well,
how did you come up with the idea to look at this species initially? Did you go through Self's material in his books first and come up with this idea? I remember when you were coming up here, but did you decide to do this first, or what was your impetus for going at Nematobothrium?
well, that's a good question. I was actually this whole idea of a book came up with a discussion with Ted Pardee and Jack Morris. We were having lunch and talking about our research and what we had learned from doing our research. And I really started working on a book about people and what they had learned from doing their research, what, how they had grown and what their research gave them. And that it just seemed natural to try to pick up on Self. And I think that I don't remember how it happened, but the fact that you mentioned you had that codex, that's when I said immediately I'm going to borrow it.
Yeah, look at all those.
Yeah, and that's
really,
and then, yeah. So I came back and Gabor and I went through all of the collections up there, and he pulled out those slides, but he also pulled out some specimens in alcohol. So you all have quite a bit of Nematobothrium material, and I don't know what else you may have in the way of notes or text or files. So I think the whole revisiting the Nematobothrium phenomenon would be kind of an interesting project for some serious undergrad or master's student, a museum-based project. Yeah.
Yeah, so using the collection, not only the collection, but the field notes from the collections.
Yeah.
So interesting.
And I have no idea who's whether they're still commercially fishing Lake Texoma, but if they are, then good grief. A week at UOBS would give you some specimens in alcohol for molecular work.
Yeah, that's amazing. So just to look at that writing by those students and professors,
Yeah.
that handwriting, those notes, so important, John. It's so cool. You have that information and that you were able to get through it and then interpret the codex for us to understand what's going on. Because if you didn't do it, it would be a long time in coming if you had done it,
Well, there's still about 80% of all of that stuff somebody should get in and do. I did not go in and record every fish. I did take one of those collections and make a spreadsheet of it. But there are others.
In fact, all your notes from Cheyenne Bottoms and Dr. Self's work from Cheyenne Bottoms and the virus project, all that material is there.
Yeah.
Just waiting to be mined. And it's we have to make sure we notate it now so people can get at it later. It's well saved, but I think it still needs better notation, better notes on it to say what it is.
Yeah. Yeah.
So I think it's well saved in the fireproof cabinets, but oh man, so interesting. So any other questions? I monopolized the question. Does anybody else have anything out there?
Oh,
Yeah, I have a question.
Go ahead. Is it Valeria?
Yeah.
Yes.
Thank you, John. This was amazing. I have to say that something that you said catch my attention a lot. So if I understood, you also believe that sometimes assumptions are very dangerous pitfall for when you are searching for the truth. And I really believe that this is something we have to pay attention on very carefully. But probably another pitfall is in these days where we are also looking for digitized all these notes and material we have in the museum preserved, observations, and also this need to use machine learning approach, artificial intelligence, this large language model. So what do you think about this necessity to summarize the data from notes and description that are so well written, so well understood when you take your time and read them into a searchable, concise, and precise database that has its own needs? So what do you think about it?
I think it should be done, absolutely. But how you do it and where the human resources come from to do it is an issue. So yeah. I quite frankly, I think that, for example, in the case of those codices, if you take, I know that the PDF files that, because I scanned all those things, I know that the PDF files can be read into Word, into Word pretty easily. And once they're read into Word, then all you have to do is go through and edit them. And once that, and then you could put those up, all that stuff up somewhere online. You could self-publish it, I guess, and it would then and make it free, and AI search engines would find it. That's my guess.
Yeah, I think we could actually utilize the text recognition algorithm to then read it automatically and then sort through the details and come up with something. The problem with that is it's difficult to imagine that an AI could go back and make some emails to people who are still living to try to connect with them to find out who did what in 1963.
Well, my efforts to do that were totally unsuccessful, except for Barbara Neese, and I may continue to follow up with her just a little bit, but no, everybody was dead, and the people that I tried to communicate with who were still alive and would have been contemporaries of those folks, no response.
Wow, interesting.
So they either, and I don't know why.
Yeah. Well, maybe they weren't mostly academics for the rest of their lives and they didn't do that. Who knows? But, we have reams and reams of field notes from all of our field trips and all of our work everywhere in the world. And so, that stuff's just sitting there waiting for us. And we use them every day here. It's pretty interesting to know that.
Yeah.
So, all right. Any other questions or comments from anywhere? You can go ahead and unmute and start talking or send a text. One of the most, this is like, I don't know. Sometimes I watch a talk and I'm interested, but this one was just like making me want to do more of this stuff. So really, really good.
It will consume your time in a hurry.
Yes. In fact, I'm working on this Galapagos paper right now, and I right now have in front of me 1889 paper. So I'm going through the paper to see what they have to say about parasites from Galapagos birds and mammals, and not much, hardly anybody. I don't think any parasites were ever collected from any of the so-called finches, which are actually tanagers of the Galapagos Islands. So one time, one person, Peter Grant, mentioned that he saw a segment of a tapeworm in some feces from a finch, and that was it. Anyway, okay, so we have all those material here. What we need to do is write out a short index card saying what needs to be done And when the students come in from parasitology who want to do some work, I can say, this is a project. It'll take you 3 years. So, all right. I think that would be good. Why don't you write out that project on an index card, give it to me, and then when the students come in here, which they're starting to come in now, we can then suggest this as one project. So I think that would be a good one.
So I might send it to you as an email instead.
Sure, do that. I have a card, an index card that was written by my uncle when I first started in parasitology in Alaska. And if it was an email, I wouldn't have it anymore. But I have this card written in India ink. The card's turning brown, but the ink is still there. And it basically outlined my master's degree working with Jerry Schmidt.
Yeah.
So, and I found the card stuck in my thesis. All right. Any other questions? Anybody from here local got one? No? John, that was amazing.
Thanks, Scott.
And I can't wait to see the book. And I'm really excited to go to the next stage here. Basically, this is the last talk of the year, or of the school year in the spring. We will start again next fall. And I think it's about like August 26th or so that we start again. And I will send out emails and put things on Facebook, put things on Instagram. That Instagram post where I put all those tinea pictures in those brains got more interest than anything else I've ever put in there. So pretty cool. Any other comments? Let's go. Let's see. One more point here. Ida wrote a note and said, that was the most beautiful talk, John. Thank you.
Thank you all.
Can't wait to read the book. And we're all excited to see it. The cover looks really cool. So, all right. Well, let's.
Well, the cover is thanks to Sourcebook's designers. Yeah.
Nice. Yeah. Well, when will we see you again?
When will you see me?
Yeah, are you coming up here? You said you're going to come by and drop off the other codex?
Yeah, it'll probably be tomorrow when I come by with Kelly.
Yeah, so I'm going to be teaching tomorrow from 10:30 to 11:45, and I'll be back up here, and then I'll have a little time. Sue's feeling good, so I
Okay.
probably won't have to go home. Things are calming down at home. So,
Okay, good.
all right. See y'all later. Thanks very much.
Okay.
It was so cool.
Bye, y'all.
Thanks. Bye.
Bye.
Very nice.
Hey, Scott.
Hi, Luma.
Hi.
Did you get my email? Let me see if I can turn off the recording here.
Okay.
Grant or I know how to do
Today.
that.
Mm.
I assume
Mm.
I don't see it.
a bit more.
More? Oh, here.
There you go.